Isabel really embodies the arts and sciences as a Renaissance woman who can combine those two things, and you'll get to see that today.
We should just clap. Like Dr. Lee said, my name is Isabel Lee, and this project is the chemistry portion of my Chemistry and Art senior capstones. And if you are interested in learning more about the art portion, you should come to our senior show. It's Friday, May 3rd. It starts at 5, and we're gonna have fancy bread. So that would entice you enough. So something that I really love about both science and art is the ability to always be learning new techniques and discovering new things about each of them. And so I really wanted to embody that in this project. And so this is going to be kind of an experimental journey of me going through and learning as much as I can using the techniques that I have gained through my biochemical education here at WLC. So the project that I decided to do was to look into the science of dyeing yarn. I really enjoy yarn crafts, and I had never tried to hand dye yarn before. So I was able to purchase dyes from a commercial seller. However, because it's a proprietary product, I didn't get any chemical information or anything that could be found online. And so I thought it would be cool to start from scratch and see and just figure out how much I can learn. And as any good science or art project begins, you have to start with a literature search. So I was able to find out some preliminary information and learn a lot more about this kind of combination of art and science. So the first thing I learned was that there are a lot of different types of dyes used for yarn, and I purchased 2 for this project. The first type were acid dyes, and these are the colors that I got them in. And acid dyes are used to dye protein-based yarns, and so the yarns that I had were silk and wool for this project. And so a little more in the chemistry of it. Acid dyes are traditionally sulfonic acids or possibly carboxylic acids. And so the protein fibers of the yarn are made up of amino acid groups, which is what all proteins are made up of in humans and in animals. And so these acid dyes can form an ionic bond with the amino acids in the protein, causing a very strong bond, and it prevents dye from being washed out after the dye process is done. And the other type of dye that I purchased were fiber reactive dyes. Again, these are the fun color names. And these are for dyeing cellulose-based yarns. So I had bamboo and cotton yarns for my project. And these are the only type of fiber dyes that contain a reactive group. And so this reactive group will react with the cellulose, which makes up these plant-based yarns, and it actually forms a covalent bond with a hydroxyl group in the cellulose. So again, this is a very strong chemical bond, and it prevents the color from being washed out, which is something called color fast in the industry of dyeing yarn. So the 2 types of dyes that I purchased were very color fast, but I wanted to know more than this. This is just available online, and I had no way of really knowing that this is actually what the specific dyes that I bought did. This is just kind of general information. So I performed several experiments. I'm going to go through many of them in briefly, and if you're interested to learn more, I do have a website. It's just isabel-lee.org that I made for this project. I recommend looking at it on a computer. It doesn't look great on mobile, so that you can see a lot more in detail all of these experiments. But the first one I performed is UV-Vis, and so this stands for ultraviolet visible spectroscopy. I'm sure you guys have seen an image like this before. This is the color wavelength spectra that we can see. And what UV-Vis does is it detects what wavelengths of light are absorbed or reflected from a sample. So this is an example spectra that I've obtained of my dye wisteria purple. And we can see that the dye does not absorb light in the specific regions of the blue and red wavelengths, which is really cool to just kind of see in more of a quantitative way why this purple is the color we see it is. And then I wanted to do some more testing, and I did two types of separation experiments. So the first one is TLC, stands for thin layer chromatography. This is a simple chemical assay that separates molecules based on polarity, which is an intrinsic kind of aspect of a chemical, and also gel electrophoresis. Gel electrophoresis is actually a biology technique, and for my literature, I wasn't able to find anybody using it for anything other than biology, but how it works is you make a gel-like matrix, and so then you run an electrical charge through it, and since DNA has a negative charge, it kind of gets pulled along through the matrix, and bigger things can't go as far because they get stuck, and smaller things can weave their way through and go farther. But again, to my knowledge, no one has used this for anything other than DNA, but since I knew that my dye molecules would have a charge in order to form those ionic and covalent bonds, I wanted to see what would happen. And I got surprisingly very good results for doing something that I've never tried before. And so this is the TLC, and this is the gel electrophoresis. And I just wanted to highlight the dye that I think had the most separation was my aqua green dye, and so it's labeled AG in this lane here and in this lane. And we can see on the TLC it had a huge separation, and on the gel it still has a separation. And so this tells me that there has to be two distinct molecules, one that is yellow and one that is blue, that's making up this green dye. And I can say that the yellow molecule has to be smaller than the blue one, and it has to be more polar than the blue one, which is cool information that I didn't know before. And so all of my dyes had interesting separation, and again, you can see more about that online. And so I want to spend the most time on NMR. So that stands for nuclear magnetic resonance spectroscopy, and I kind of fell in love with NMR ever since I took OChem as a freshman. And we are so blessed at WLC to have an NMR that is available for students to do research. And so I have really enjoyed my time working with it, and it's very complicated, but essentially it manipulates the hydrogens, so when you put a sample in the NMR, every individual atom has nuclei that have some sort of magnetic spin. And so the NMR can apply a magnetic field to it and manipulate that magnetic spin, and then it produces spectra, which I can interpret to discover the chemical structure of a molecule. It's very similar to how MRIs work in the healthcare industry. And so I will go into more detail, but I just want to kind of show off my spectra. And even if you've never seen an NMR spectra before, these are really good. You should be very impressed. Well, this is a proton spectra, and so each one of these peaks indicates a unique hydrogen in the molecule. And then I also got a proton spectra of my aqua green. So both of my acid dyes produced very nice spectra. My fiber reactive dyes didn't. However, you can see that they are similar but have some differences, which is interesting to see. And then I also ran a carbon NMR and a depth spectra, which looks at the carbons in the molecule. And so this is the robin egg blue spectra. And then this is the aqua green spectra, and I want to talk more about this. So, like I mentioned at the beginning, I didn't have any way of knowing if this was a carboxylic acid or a sulfonic acid based dye. And so looking at this is called a depth NMR spectra. Essentially, it's just a fancy carbon NMR. And so I was able to determine that I think it's sulfonic acid in my dye, and there are a couple peaks that indicate that. However, nobody does NMR on these types of molecules, so it's really hard for me to find literature references. But I did, and I do know that nitrogen and sulfur have similar properties on NMR, and an NO2 group has peaks around the same region where I am seeing peaks. And so I know there's no nitrogen in this sample because nitrogen can be damaging to protein-based yarns. So if they wanted to sell a good product, I don't know why they would put nitrogen in their dye. However, I also found some research that mentioned that there could be carbons in the 50 to 60 ppm range. And this peak is actually really cool. As you can see, it's the only one that's going down. And I've only ever seen this in like textbooks, and so like I got this. This is mine. I'm gonna frame it once I'm done with this project. And so in depth NMR, carbons that form peaks going down have to be CH2s. So I know that there's at least two CH2s in my molecule, which is really, really cool to know. Again, I have more references on my website if you're interested in that. And so for my conclusion, I kind of started off with a vague idea of just like, I want to learn as much as I can. And I started with very little information, but I was able to discover partial chemical structure, different polarities, as well as the molecular size and charge of my different dyes. And this didn't change how I dyed my yarn or how I went about my art, but it does give me a greater insight into the significant aspect of my art. And I think that we should continue encouraging people to just do science out of curiosity. You don't need to be saving the world or doing cancer research to make your scientific journey significant. And now I just know more about the dyes that I have used, and that makes me happy, and that's great. And I also, it was really great to use all of the techniques that I've learned at WLC to do something that I've never done before. In like labs, we always have an unknown that we're testing, but somebody knows it. And so this is a true unknown, and so that was really fun for me. And I just like to thank WLC and WLC's NMR my research, as well as Dr. Dan Abling, who was my mentor on this, and Jeremy Vanderheide, who bought my things, as well as Dr. Jill Davis and Dr. John Warner, who gave me some helpful insight into doing this project. Thank you so much.
Thank you. Very interesting, and we have probably 3 or 4 minutes for questions. If anybody has questions or comments.
So the NMR is really cool. But I do phosphorus NMR, so the carbon and hydrogen stuff I don't know as much about. But you had 2 side-by-side NMR scans. What were? What was the left and what was the right? Like what was the difference?
So for the proton, it's just a zoomed in version. So you can see this is the full spectrum. This is just zoomed in on the peaks. And then for the carbon, so this is just a carbon NMR, and this is the depth carbon NMR. So like the special carbon NMR.
I was gonna ask, could you use this information to possibly develop your own colors and dyes?
it would definitely be a good start. There's a lot more that I would want to look into if I wanted to do that. Synthesizing dyes is definitely something that I'm interested in, and it would more be like if I were to make my own dyes, these would be validation techniques that I did actually make what I set out to make.
Did someone else have a question?
So for someone who has no idea how to read these NMR results, what makes it good? How can I look at this and like, what are you looking for in results that tells you information? Separation of spikes?
Yeah,
size,
sure. So you can see on this NMR, there's a lot of like, call it grass. And so it's a little bit difficult to actually tell where distinct peaks are. So like these are kind of probably peaks, but there might be some small ones that are lost. And so when running depth, it actually, you can see, reduces that significantly. So like this little small peak that we might not have seen on that one can be seen. And so it's kind of considered a good NMR if you have very clear peaks. Also if there is like a kind of, not all of them are the same height. So the heights are actually significant. And if they were all the same height, that would just be like kind of suspicious. The height is correlated to actually, so the integral of the peak determines how many of that molecule make up the peak. So since this is carbon NMR, if you took the integration of all of these, that would be a ratio of how many carbons are in there. So if they were all the same size, that would be kind of suspicious. And so, I mean, you can also just believe me, that doesn't go. But so, just like the very clean, nice, kind of not super lost in the noise is a big indication. And a lot of times if you have like a contaminant in your sample, it's just gonna be like all peaks. Like the kind of this region gets a little bit messier, and if you can imagine just the entire spectra like that, because it's detecting different things. And so the fact that I have really nice, distinct, and a clean spectra is really great. Yeah.
Well, seeing no other hands, we'll go ahead and thank Isabel again. We will have 4 or 5 minutes, we can transition